begin the lecture about antiviral drugs. If you remember from microbiology, viruses are very small infective particles. Which contain DNA or RNA molecules which are enclosed in nucleocapsid. In protein envelope as a whole, that is called nucleocapsid. Some viruses also have lipoprotein envelope, on the surface of which there are some glycoproteins which serve as antigens. As viruses may have DNA or RNA molecules, they are divided in two. DNA viruses, among them, you must pay more attention to herpes viruses, which cause chickenpox, shingles, cold sores, glandular fever, because there are a lot of treatments nowadays against herpes viruses. RNA viruses, they include a lot of viruses. Among them, there are viruses which cause influenza, and also appropriate treatment, specific treatment is available for retroviruses, HIV, which causes AIDS, acquired immunodeficiency syndrome, and also a lot of drugs nowadays are available against this disease, this infection. Hepatinoviruses, hepatitis cause they, especially hepatitis C, and also a lot of drugs are available. Nowadays, more new drugs have been introduced into clinical practice against hepatitis C viruses. And also coronaviruses, so very bad virus, which is so famous now, also belong to RNA viruses. Among viruses, as I said before, there are such against some treatment is available. Not so many viruses respond to antiviral drugs, such as herpes viruses, among them herpes simplex, varicella zoster, Epstein-Barr virus, and cytomegalovirus, HIV, human immunodeficiency virus, influenza types and B, respiratory syncytial virus, and hepatitis viruses C and B. Some others, a little bit. It is necessary to remember that antiviral drugs share the common property of being virus-stated, not virucidal. And it is very important they are active only against replicating viruses and don't affect latent viruses, latent forms of them. Therefore, some viruses may live in human organism for a long time in latent forms, such as herpes viruses and also HIV. HIV may live in latent form several years and don't cause any symptoms, but at some days they are evoked and cause inhibition of immune response, dramatic inhibition of immune response. So it's necessary at first refresh your memory, I think that you know it from microbiology, or recognize about how life cycle is provided. In summary, nearly all viruses replicate via the following stages: attachment and entry, uncoating, gene expression and genome replication, assembly and egress. Some viruses have additional stages such as maturation, it is common for HIV virus, and release, it is common for influenza virus. This picture represents how viruses replicate. So the first step, as I said before, attachment. Viruses have special proteins on their surface. It may be proteins on the envelope or on protein coat, and through which viruses bind to normal constituents of host cell membrane. It may be different receptors for neurotransmitters, hormones, cytokines, ion channels. I give you some examples. Rabies viruses, viruses which cause rabies, bind to the nicotinic receptors on the surface of skeletal muscle. Therefore, one of the symptoms of this infection is dramatic paralysis of skeletal muscle, among other symptoms, of course. And it is very well known that HIV binds to CD4 proteins on T lymphocytes. T lymphocytes, they have special CD4 proteins and chemokine receptors. So they, of course, exist for cytokines, not for viruses. But a virus have ability to bind to these proteins, enter T lymphocytes, and cause dramatic suppression of immune response. And therefore, such patients can suffer from different infections, including Pneumocystis carinii pneumonia, cytomegaloviral infection, aspergillosis, such infections which commonly don't present in patients, in individuals with normal immune system function. Yeah, so let's go on. After attachments, viruses enter the cell. During entry, they may remove their coat, or they remove this coat inside. It will depend on the type of viruses. So after uncoating, it is called uncoating, this removal of the coat, viruses replicate. So viral genome is reproduced. It may be outside in cytoplasm for RNA viruses, and it may be inside, especially for DNA viruses, and such RNA virus is influenza virus, inside nucleus. After replication of genome, viruses can translate messenger RNA into some viral proteins. Commonly, it is structural proteins, but it may be some enzymes, viral enzymes, which help them to replicate. It's necessary to remember that after structural proteins synthesis, assembly of new variants occur. So these proteins may be around the core, can form the code or envelope, structural proteins. And after variants are formed, they go out from the cell, egress. They may egress by budding or by lysis. In the case of lysis, host cell, of course, is destroyed. So you may see here what I said before. And then it's necessary to differentiate how DNA viruses and RNA viruses replicate. DNA viruses, as I said before, replicate inside nucleus. So DNA is transcribed into messenger RNA, and messenger RNA is translated into proteins. These proteins may be viral DNA, viral DNA polymerase, which is used for the synthesis of new viral DNA. Or it may be such proteins which form capsid and envelope, that is, structural proteins. After assembly, viruses new viral particles, they aggress by budding mainly. So you may read about how does it occur, what I said you before. So after assembly of code proteins around the viral DNA, complete variants are released by budding or after cell lysis. This picture represents RNA virus which enter the cell by endocytosis. This is an example of influenza virus. We will speak about it more closely a little bit later, but now look that in this case RNA replication must be. So RNA replicate using host cell enzyme RNA polymerase. In influenza virus, it does occur inside nucleus. In some other RNA viruses, it may be outside, in cytoplasm. So viral RNA is synthesized, and also messenger RNA is synthesized. Messenger RNA is used for the synthesis of structural proteins. So it's transcribed, is translated into structural proteins. And after assembly, this virus aggress by budding. This is text which mimics what I said before. And now let's look at the classification of antiviral drugs according to mechanism of action. That is, according, depending on at which step of viral replication, viral life cycle they act on. At first, inhibitors of viral attachment and entry. It is at first gamma globulin. Pooled globulin we will speak a little bit later about. The same is about monoclonal antibodies against respiratory syncytial virus called polivizumab. Then chemokine receptor blocker maravirok and inhibitors of fusion of viral and host cell membrane and enfuvirtide. So maravirok is inhibitor of attachment and enfuvirtide is inhibitor of entry into HIV. So these two drugs are effective against HIV. Then inhibitors of viral encoding. We have only rimantadine and amantadine, such drugs which are effective against influenza A virus. Then inhibitors of viral genome replication. This group includes a lot of antiviral drugs against different infections. For example, inhibitors of viral DNA synthesis, DNA polymerase inhibitors. Of course, they are directed against DNA viruses, and as I said before, it is the only virus, herpes simplex, against the specific treatment is available. So anti-herpes virus nucleoside and nucleotide analogs. Among them, the main is cyclovir, we will speak about a little bit later. Then, very important drug, ganciclovir and cidofovir, which are important, act against cytomegaloviruses. You have some questions?
Yes, there are some people, they just want to join. Can you please join them?
What?
Some students, they're trying to join, but you need to let them in.
Так, сейчас.
Наверху у вас, Дилара Ахметовна.
Где?
Наверху участники опять. Принять.
Да, все. Угу. Так, принять. Ну что? Вошли, нет?
Да, вошли.
Да, ну все, да, можно продолжать, да? Угу. Так. Дальше. Значит, we are speaking about anti-herpes nucleosides and nucleotide analogs. Yeah? A little bit about drugs such as idoxuridine and trifluridine. You see that they are useful against keratitis caused by herpes simplex viruses, only for local use, application in the eyes. Then, besides nucleoside analogs, we have non-nucleosides, such as foscarnet. We are speaking a little bit later. And now about such drugs which are effective against HIV. Against HIV enzyme, which is called reverse transcriptase, and really it is RNA-dependent DNA polymerase. Therefore, we are speaking about in this group, in this classification about this group. Then HIV integrase inhibitors, then inhibitors of viral RNA synthesis, not DNA, pay attention, RNA synthesis, against hepatitis C viruses. Among them also there are nucleosides, RNA polymerase inhibitors, and non-nucleosides, RNA polymerase inhibitors. And unique group of such viruses against this component of viral particles. Yeah, then inhibitors of viral maturation, it is group unique only for treatment of HIV. Then inhibitors of viral release, it is group only for the treatment of influenza infections A and B. And some other antiviral drugs with different mechanisms of action we will speak about. So now we are speaking about large group which inhibit DNA polymerase. As a whole, and polymerases are DNA or RNA polymerases as a whole. The vast majority of drugs that inhibit viral genome replication inhibit polymerase. Most viruses encode their own polymerases. These enzymes are required to replicate the viral genomes and differ in various ways from human polymerases, making them excellent targets for antiviral drugs. That is, what is important to understand, that these viral polymerases differ from human cell polymerases, and therefore they may serve as target for antiviral drug action. So these antiviral drugs act mainly on viral polymerases, then on human polymerases, human cell polymerases. Viruses expressing polymerases that have been successfully targeted, they include human herpes viruses, HIV, hepatitis B viruses, and hepatitis C viruses. Most of these drugs are so-called nucleoside analogs. But also some non-nucleosides are available, we are speaking about. Yeah, and now about nucleoside analogs which inhibit polymerases. They have in common the following characteristics. These nucleoside analogs must be phosphorylated to triphosphates. In this nucleoside analogs, triphosphates, incorporated into DNA chain instead of normal nucleosides, which are normal constituents of DNA. Yeah, so you see, the common features of enzyme inhibition and incorporation into DNA or RNA can be important for antiviral activity. Selectivity of antiviral drugs against viruses depends on how much more more viral enzyme phosphorylate the drug than cellular enzymes do. So you must understand, as I said before, that these drugs are phosphorylated to triphosphates, and enzymes which phosphorylate these drugs to monophosphate at first, then diphosphate and triphosphate, they must be more susceptible to viruses than enzymes which phosphorylate human cell DNA. Then DNA polymerase or RNA polymerase must be more susceptible in viruses, so viral DNA polymerase and RNA polymerases must be more susceptible to these drugs than human cell polymerases. And now we are speaking about herpes viruses, treatment of diseases caused by herpes viruses. They include, as I said before, herpes simplex that can cause cold sores, conjunctivitis, mouth ulcer, genital infections is caused by herpes simplex type 2, and rarely but very seriously encephalitis. But this commonly develops only in immunocompromised patients. And of course, it is more aggressive disease. Then varicella zoster virus causes shingles and chickenpox in children and sometimes in adults. Epstein-Barr virus causes glandular fever. And about cytomegalovirus, it is the most dangerous virus among herpes viruses. So it can cause very severe diseases in patients, especially in the setting of immunosuppression. Immunosuppression may occur in patients with AIDS or after administration of immunosuppressants, for example, during treatment in the case of organ transplantation. Dissemination of infection results in end organ disease, including retinitis, which can result in loss of vision, colitis, esophagitis, central nervous system disease, and pneumonitis. And cytomegalovirus can affect the fetus with catastrophic consequences. So if pregnant women are infected by cytomegalovirus, it can lead to death of fetus. This picture represents how herpes simplex virus looks like. You may see that DNA is enclosed in protein coat. As a whole, it is called nucleocapsid. Glycoprotein envelope also is present, and lipoprotein, envelope is present, and glycoproteins, they are present also on the surface of envelope. So herpes viruses replication is regulated multi-step process. But better I will explain using the picture. So after attachment and entry, a virus as usually undergoes encoding. It is interesting that herpes viruses also loses its code when it enters the nucleus. It must obligatory enter the nucleus because DNA is transcribed into RNA inside nucleus. It is called early transcription because this messenger RNA, which are found, are translated into viral protein called DNA polymerase. So such viral DNA polymerase, which is needed for synthesis of new viral DNA molecules, that is, for reproduction of viral genome. After viral genome is reproduced, delayed transcription does occur, that is, this viral DNA is transcribed to messenger RNA and then is translated into proteins which are called structural proteins, which are needed for formation of coat and envelope. And after assembly, a viral goes out by budding. So what is the target for drug action? Antiviral drug action here, of course, viral DNA polymerase. And here you can see the drugs which belong to this group, nucleoside and nucleotide analogs. It's necessary to say that acyclovir and all others belong to nucleoside analogs, and only cidofovir belongs to nucleotide analogs. Why is it so? I will show you a little bit later using pictures. And also non-nucleoside drug is available for the treatment of herpes simplex infections. Is called foscarnet. About acyclovir. It is a guanosine derivative with a high specificity for herpes simplex and varicella zoster viruses. So it is highly effective against infections caused by these viruses. And you see that herpes simplex are more susceptible to acyclovir than varicella zoster. Epstein-Barr virus only slightly sensitive, and acyclovir is not effective against cytomegalovirus. You may look at the picture which represents acyclovir here, and this is desoxyguanosine derivative, which is normal constituents of DNA molecule. What is the difference between them? Acyclovir is a cyclic nucleoside, so this cycle is broken, in the structure of acyclovir. And therefore, it inhibits DNA synthesis. How does it occur? Let's look. So, acyclovir is converted to the monophosphate by thymidine kinase, the virus-specific form of this enzyme. What does it mean? Look, acyclovir is phosphorylated at first step to acyclovir monophosphate, but viral enzyme. Therefore, it is phosphorylated mainly in virally infected cells, not in other human cells. It is very important and remembered. But then it is phosphorylated by cellular kinases to acyclovir diphosphate at first and acyclovir triphosphate. But at first step, remember, it is phosphorylated by using viral enzyme, and therefore this step is very specific for virally infected cells. Then let's look how does occur. This picture is better to show you how acyclovir triphosphate at first competes with desoxyguanosine triphosphate, the normal nucleoside which is incorporated into DNA chain. So it displaces it and is incorporated into DNA chain instead of. And it terminates chain growth. So DNA chain doesn't grow more. It also leads, this incorporation, to the so-called inhibition of DNA polymerase. So when the next desoxynucleoside triphosphate binds, viral DNA polymerase is frozen. How does it occur? The matter is that the polymerase translocates to the next position on the template, but cannot add a new desoxyribonucleoside triphosphate because there is no 3-hydroxyl on acyclovir triphosphate. Let's look at the picture, this picture. You see that normal nucleoside has 3 hydroxyl groups, but acyclovir has only 1. And therefore, after incorporation of this acyclovir triphosphate, what will happen? The viral polymerase freezes in a so-called dead end complex. Why does it occur? It is not known well. It is not recognized completely. What is necessary to know about acyclovir? Acyclovir is 30 times more potent against the herpes virus enzyme than against the host enzyme. It is very important for selectivity of acyclovir against viruses if you compare it with human cells, host cells. And then acyclovir triphosphate is rapidly broken down within the host cell by cell phosphatases. So it's present in human cells not so long. So it is very quickly removed from the cells by phosphatases. So all these reasons are responsible for low toxicity of acyclovir, which are 3 reasons. The first, it is what I said before, that acyclovir is phosphorylated by viral thymidine kinase at the first step. Then, what you can see here, acyclovir is 30 times more potent against herpes virus DNA polymerase than human cell DNA polymerase. It is second reason. And third reason, that it is removed from the cells, human cells, very quickly. It is treatment diseases in which acyclovir is used. You may read it. I won't pronounce it for you. You may read. Then, but acyclovir, it may be used, of course, by oral route. But during oral route, it is bioavailability is not so high. Therefore, prodrugs such as valacyclovir has been introduced into clinical practice, which releases, of course, acyclovir, rapidly converted to when ingested orally. But in the plasma, it is converted to acyclovir, and bioavailability, in this case, 3 to 5 times greater than bioavailability of acyclovir itself. The same is for another drug, penciclovir, which has the same properties and mechanism of action as acyclovir, but acts a little bit longer. It also has prodrug from cyclovir, which has higher bioavailability. And here you may look at the pictures which represent the structures of acyclovir. You see, this is acyclovir, but I showed you desoxyguanosine. Acyclovir displaces it from DNA strand, competes with. You may see that it is cycle and this is acyclic. Then valacyclovir prodrug. And about ganciclovir, let's look. Ganciclovir has 2 hydroxyl groups, unlike acyclovir. But it also acyclic, So it differs from normal desoxyguanosine. How does it act? The mechanism is the same as for acyclovir. But what is the difference? The matter is that ganciclovir is more toxic than acyclovir. So it has the difference between sensitivity of DNA polymerase, viral DNA polymerase, and human cell polymerase is not so high as for acyclovir. So cytomegalovirus is inhibited, DNA polymerase is inhibited almost at the same concentrations as DNA polymerase in human cells. Maybe a little bit higher must be such concentrations, but the difference is not so large as for acyclovir. And then ganciclovir more closely resembles the natural desoxyguanosine, and this resemblance may account for its greater toxicity. You may read it. Ganciclovir, of course, may be used also against herpes simplex and varicella zoster viruses. But it is not optimal treatment here because acyclovir also is very effective and much less toxic. So what is the function of ganciclovir in the treatment of herpes simplex infection? The matter is that it is very effective against cytomegaloviruses, cytomegaloviral infections. So it's necessary to say that cytomegaloviruses, they have no thymidine kinase as herpes simplex and varicella zoster, but they have this enzyme, phosphotransferase UL97, which also can phosphorylate ganciclovir to monophosphate. This enzyme cannot phosphorylate acyclovir to monophosphate, only ganciclovir. So you see that therefore ganciclovir, unlike acyclovir, is effective against cytomegaloviruses. So toxicity of ganciclovir depends on that DNA polymerase, viral DNA polymerase, is sensitive, but human DNA polymerase also is sensitive to more extent than to acyclovir. Especially such DNA polymerase, which is located in mitochondria, is more sensitive of human cells. And then you may read also ganciclovir, it is not rapidly broken down as acyclovir in human cells. It has been shown to persist in cells infected with cytomegalovirus for up to 20 hours. It also may be the reason of higher toxicity of ganciclovir. What is the common toxicity of ganciclovir? It is very well understandable. Yeah, we spoke about when we discussed immunosuppressive drugs. They also can suppress DNA synthesis, and suppression of DNA synthesis in human cells suppress mainly division of such cells which are divided rapidly. Which cells are divided rapidly? Of course, bone marrow cells. And therefore, ganciclovir mainly inhibits division of bone marrow cells and can cause neutropenia. It is common adverse effect of this drug. And now about cidofovir. Let's look at the picture I showed you before. Look at this. It is cidofovir. What is the difference between ganciclovir? It is phosphorylated at the first step. So, if some viruses acquire resistance, cytomegaloviruses, of course, acquire resistance to ganciclovir because they lack of this enzyme which can phosphorylate ganciclovir at the first step, this drug cidofovir may be effective because it already has this phosphate group. It is already monophosphate. So you may read that cidofovir is effective against such strains of cytomegaloviruses which are resistant to ganciclovir. You may read it. But cidofovir is used only intravenously. Yeah, and now about non-nucleoside analog foscarnet. You see that all nucleosides must be phosphorylated, and therefore resistance may develop if these viruses lose their ability to phosphorylate these drugs at the first step. In this case, we are speaking about ganciclovir. But in this case, foscarnet may be useful, may be effective, because it doesn't require phosphorylation at all. May read about it. Foscarnet has a relatively broad spectrum of activity in vitro. It also is effective against HIV, but clinically it is used to treat only certain infections caused by herpes simplex and cytomegaloviruses. In the case if acyclovir or ganciclovir are ineffective because of development of resistance of these viruses to these drugs. So, mechanism of action, of course, different from nucleoside analogs. Doesn't require activation, but it inhibits viral DNA polymerase directly by mimicking the phosphonate product of DNA polymerization. Let's go to another virus, which is called HIV, human immunodeficiency virus. About structure of this virus, I will show you the picture at first. You see, this is this virus. What does it have? It has envelope which contains some glycoproteins on the surface through which this virus can bind to membrane of T lymphocytes. This is T lymphocyte. Besides, it has capsid, protein coat, and has 3 enzymes, viral enzymes: integrase, reverse transcriptase, and protease. What is the function of these enzymes? They function such as reverse transcriptase, integrase. They help this virus to replicate, to reproduce its genome, and protease helps to this virus to maturate. This step is called maturation. That is to be effective against. So you see, when virus enters the cell, how does it enter the cell? It binds to CD4 proteins on the surface through glycoprotein 120, and also can bind to chemokine receptor. Otherwise, if it doesn't bind to chemokine receptor, it doesn't enter inside. Then fusion of human cell membrane, that is lymphocyte membrane, with viral envelope does occur. And after that, uncoating, that is removal of protein coat, occurs inside cytoplasm. And the next step is unique for the so-called retroviruses. So it is RNA virus, but it synthesizes on DNA molecule by enzyme which is called reverse transcriptase. So it is reverse transcription of viral RNA to viral DNA. Then this viral DNA, which is synthesized, must be introduced into human genome in the nucleus. And for this, the special enzyme viruses have, which is called integrase. So this integrase helps this DNA provirus, it is called this DNA provirus, to be inserted inside human DNA. And what will happen next? In this case, this provirus makes human DNA molecule to be transcribed into viral RNA molecules. Viral RNA may be of 2 types: genomic RNA and messenger RNA. Genomic RNA, it is evident here, RNA which represents the viral genome, which is enclosed in protein coat. Messenger RNA is needed for the synthesis of viral protein. At first, long-chain viral protein is formed. And then virus with this long-chain protein goes out from the cell. And outside lymphocyte, it undergoes the step which is called maturation. What does it mean? You see, viral protease cleaves this long-chain protein into some blocks, some short proteins, and these proteins may form this protein coat around this core of virus. And as I said before, this step occurs outside. It is called maturation. So protease provides maturation of viruses. They become completely effective against other human cells. And this picture represents the same, but with some antiviral drugs groups which are used against HIV. We have such drugs which block this chemakin receptor. It is called maraviroc, and binds to chemakin proteins on surface of membrane of lymphocytes, prevent attachment. Then what may be blocked also? Fusion inhibitors, such which inhibit fusion of envelope of viruses with human membrane of lymphocyte membrane. This drug is called enfibertide. Then we have a lot of drugs which inhibit reverse transcription. Among them, there are nucleoside reverse transcriptase inhibitors and non-nucleoside reverse transcriptase inhibitors. Then enzymes integrase also has special antiviral drugs against it. Inhibitors of integration. And at last, inhibitors of protease. So you see, and therefore inhibitors of maturation. So you can see that all these viral enzymes have some inhibitors which may be used as antiviral drugs against HIV infections. And besides, some drugs are available which inhibit attachment and entry. So here you may see drugs, nucleoside inhibitors of reverse transcriptase. The first was azidothymidine, which also is called zidovudine. And some others. What is the mechanism of? Mechanism is common for all such nucleoside inhibitors of DNA polymerase. You must remember, and understand that reverse transcriptase is DNA polymerase, but RNA dependent. So I would like to represent you drugs which inhibit reverse transcriptases. But at first, of course, we are speaking about zidovudine. It is an analog of thymidine. Instead of normal desoxythymidine, it is incorporated into DNA molecule instead of. But at first, it must be, of course, phosphorylated. But it is interesting, and remember that zidovudine is phosphorylated completely by cellular kinases. That is, it has no specificity or selectivity for such cells which are infected by viruses. So it may be phosphorylated also in human cells. Therefore, zidovudine is not so safe as acyclovir and even goncyclovir. So triphosphates then also compete with normal desoxythymidine and is incorporated instead of into growing DNA strand and causes chain termination. That is always is completely the same as for acyclovir. Then it's necessary to understand that mammalian alpha DNA polymerase is relatively resistant to this drug, But gamma DNA polymerase, which is present in mitochondria, is more sensitive to this drug, and therefore it may lead to adverse effects. Which adverse effects are common for, how do you mean? The same for ganciclovir, that is inhibition of bone marrow, suppression of bone marrow, and therefore the main adverse effect is neutropenia and anemia. So necessary to remember that zidovudine is more toxic than acyclovir and even ganciclovir. Then I will show you the picture, not this picture. Let's go back. Pay attention to this drug, tenofovir. What can you see here? What is the difference between this drug and other nucleoside analogs of anti-herpes simplex analogs of given here? It is already phosphorylated at the first step, not only at the first step. So it may be effective against such strains of HIV which acquire resistance because they lack phosphorylated enzymes. So tenofovir is more effective against resistant strains. But against resistant strains also non-nucleoside reverse transcriptase inhibitors are available, such as efavirenz and others. They are more effective against HIV-1 than HIV-2 reverse transcriptase, but may be effective against such strains which are resistant to nucleosides. Foscarnet also is given here because I said you before that foscarnet also is somewhat effective against HIV, not only against herpes viruses, cytomegaloviruses. Also may be used if it is needed in combination against HIV. This is structure of foscarnet you may see here. Phosphonate group are present, and structure of other non-nucleosides. And about inhibitors of integrase. We discussed it there that integrase provides integration of HIV genome into human DNA in nucleus. And therefore, it helps HIV genome to replicate. You may read how does integrase act here in more details, yeah? But which compounds are effective against? It is raltegravir, elvitegravir, and dolutegravir. Now three drugs are effective against integrase. And now a little bit about inhibitors of attachment and entry. So these drugs prevent infection at the first step. So it is very useful drugs for the treatment of and prevention of infection. So these drugs, they don't enter the cell, lymphocytes, they act outside. About maraviroc, which inhibits attachment. So it targets the chemokine receptor of this type, CCR5, which a host plasma membrane protein. It is interesting that development of maraviroc stemmed from clinical studies of individuals who had been exposed relatively repeatedly to HIV yet did not develop AIDS. So there are such individuals who have no such receptors on chemokine receptors on T lymphocytes, and such individuals, they are not infected by HIV-1. And then on the basis of this knowledge, this drug was designed and introduced into clinical practice. Then, about enfavertide. What is necessary to know about? Let's look at this picture. You see, this is CD4 to which virus binds through glycoprotein 120 and also to chemokine receptor. Glycoprotein 41 is needed for fusion. How does it occur? Fusion of envelope of virus and human cell membrane. So this glycoprotein 41 has two the so-called heptide regions 1 and 2. And normally, a heptide repeated region 1 and the second heptide repeated region, which is mimicked by enfavertide, they interact with each other. The glycoprotein 41 refolds so that the heptide region 2 segments bind directly to the heptide region 1 segments. This refolding brings the virion envelope and the cell membrane into close proximity, allowing membrane fusion to occur. So how it is shown here? You see, this is refolding, and this is fusion of membrane, human membrane, and envelope. But if enfavertide, which mimics heptide region 2, displaces it and is incorporated instead of this heptide region 2, this refolding doesn't occur and fusion of membrane is inhibited. So it prevents entry of virus inside cell. So these two drugs, of course, are used in combination with other anti-HIV drugs, but they, as I said before, prevent infection at the first early steps. And now about protease inhibitors. You may read that proteases is common enzymes of viruses, which provide maturation of these viruses. Several protease inhibitors have now been developed, and they are used in combination with reverse transcriptase inhibitors. It is very important for the treatment of AIDS. Example of this drugs. They may be also used in combination. Combined lopinavir-ritonavir, brand name Kaletra, is highly used nowadays, widely used for the treatment of HIV infection. Common side effects, they are common for all protease inhibitors you may read about. It's necessary to say that HIV infection, AIDS, is not treated by one or even two drugs. Several drugs are needed for effective treatment of this disease. And here, very effective combination of these drugs is given, is represented. You may see that dolutegravir, if you remember, it is inhibitor of integrase. And entricitabine is inhibitor of reverse transcriptase. Tenofovir also is inhibitor of reverse transcriptase, but effective against resistant strains, because it is phosphorylated nucleotide. It is salt of this tenofovir. So this combination is designated in the following way. You may meet it in reference books. You may also know that not only HIV infection is treated by this combination, but tenofovir also are used in patients with chronic hepatitis B viral infection. With lamivudine, and the same lamivudine also is an inhibitor of reverse transcriptase. So several inhibitors of reverse transcriptases in combination of may be used against hepatitis B infections. And now let's go on to treatment of hepatitis C viral infections. You may see that a lot of drugs are available now. Inhibitors of viral RNA synthesis, because this virus is RNA virus. Among them, there are nucleoside RNA polymerase inhibitors, non-nucleoside RNA polymerase inhibitors, and inhibitors of this molecule we are speaking about now. At first, about nucleoside and nucleotide analogs, inhibitors of RNA polymerase. So you see that RNA virus encodes an RNA-dependent RNA polymerases, and at the basis of a successful treatment of HIV and herpes infections by inhibitors of DNA polymerase, RNA polymerase inhibitors was designed and introduced in clinical practice. The first of these drugs is sofosbuvir, which contains uracil. Uracil is the base in uridine, a normal nucleoside precursor of RNA. Like cidofovir, tenofovir, and adefovir, sofosbuvir contains a phosphate group, which in this case represents a phosphor. It is not simple to read it. Phosphoramidotide that is further modified with additional groups and are cleaved off in hepatocytes. Therefore, sofosbuvir is a prodrug. Then, non-nucleoside inhibitors such as dasabuvir. We read about. But the matter is that these drugs are not used as monotherapy. They are used only together with other drugs against this infection. Other drugs represent the following inhibitors. As I said you before, NS5A is a viral protein that is essential for viral genome replication also. This protein helps RNA polymerase synthesize long chain of RNA and also alter the host cell environment in order to embed viral RNA synthesis and to have roles at later stages of infection. And drugs which are effective inhibitors of this enzyme are the following: lidipasvir and ombitasvir. And a little bit about drugs which are also very unique because hepatitis C viruses have special enzyme protease. But this protease is not involved in maturation as HIV protease. It is involved in genome replication, and better to say, gene expression. The first step in gene expression is translation of the viral genome. The translation product is a so-called polyprotein that encompasses protein from multiple viral genes, and the next crucial step is herpes simplex virus gene expression is cleavage of the polyprotein into individual proteins. That is, you see that this protease is needed to cleave large polyprotein. We discussed it with HIV protease, but if in HIV protease cleaves this long chain polyprotein into structural proteins, here this polyprotein is cleaved into proteins that replicate hepatitis C virus RNA, producing new genomes. So it is the function of inhibitors of this to inhibit this step. You may see the name of these drugs. So, and as I have said you before, these drugs are not used as monotherapy alone. They are commonly used in combination with inhibitors of RNA nucleoside, RNA polymerase nucleoside and non-nucleosides, and so on. You see, this protease is called NS34A, remember it. And now let's go on to the treatment of influenza infections. In this case, we have two groups of drugs. Drugs which inhibit uncoating and drugs which inhibit release of newly formed variants from epithelial cells of upper airways. The first group includes amantadine. If you remember, amantadine, it is the drug we discussed in the CNS, drugs acting on the CNS as anti-Parkinsonian drug, which inhibit, blocks, better to say, NMDA receptors, and also inhibits release of dopamine, release of dopamine. But it is interesting that it was noticed during influenza epidemic that patients who are treated with amantadine, with Parkinson's disease, of course, are not infected by influenza viruses. Commonly, they don't fall into the disease. Didn't fall. And therefore, this amantadine was also tried for use in for the treatment of influenza. But another analog of amantadine is rimantadine, is more useful because it has better pharmacokinetic properties. Mechanism of action, let's look. This is influenza virus. It enters the cell by endocytosis. It binds at first to sialic acid on the surface of epithelial cells of upper airways through proteins which are called hemagglutinin. Then they enter the cell by endocytosis. Endosome is formed, but viral genome releases from because of the following mechanism. Let's look at this picture. It represents in more details this mechanism. So you see, this is endosome, and this is influenza virus inside. Hemagglutinin is bound to sialic acid on the membrane of this endosome. Through this mechanism, through this transporter, hydrogen ions enter the endosome. And then it does these hydrogen ions enter inside to the virus, influenza virus, through special channels. It is protein called M2. And acidification of medium inside virus leads to removal of protein coat, and genetic material is released into cytoplasm. What is the mechanism of action of amantadine and rimantadine? It is very simple. They simply block these M2 proteins. And therefore, they prevent acidification of medium inside viruses, and therefore they prevent uncoating and release of virus into cytoplasm. Release of viral genome, into cytoplasm. But unfortunately, these drugs are not so effective for the treatment of deep disease. They are effective only at the first stages of the disease, when symptoms only begin to develop, and may be used especially for prophylaxis if such patients have contact with infected individuals. And they are effective only against influenza virus type A. Then let's discuss another group. You see that after uncoating, viral RNA enters the nucleus and is replicated using viral RNA polymerase into viral RNA and messenger RNA. Messenger RNA are translated into structural proteins, then assembled as a K, and newly formed variants released from the cell. But the matter is that, as I said before, hemagglutinin binds to sialic acid residues on the surface of epithelial cells. and therefore these viral particles cannot go out. But they have special mechanism to do it. They have enzyme which is called neuraminidase, which also is present on the surface of envelope of these viruses. And this neuraminidase, they splits sialic acid, and therefore these viruses can freely go out from release from the cells. Then they can bind outside, also because hemagglutinin can bind to sialic acids residues on the epithelial cells surface. And again, neuraminidase splits the sialic acid, and these viruses can spread freely in airways and can infect other nearby cells. And drugs which inhibit neuraminidase, such as zanamivir, oseltamivir, and nirmatrelvir, they prevent this release and spread. It is interesting that these drugs resemble the structure of sialic acid. So you may read it after. You see that this is structure of sialic acid. This is structure of zanamivir. This is structure of active metabolite of oseltamivir. Oseltamivir is a prodrug. And this picture represents the chemical structures of this. You may see that zanamivir and oseltamivir resemble the structure of sialic acid. So it is design of drugs which has some modern features, possible to say. What is necessary to say about zanamivir and oseltamivir? Zanamivir is given only by inhalation. And therefore, it may cause bronchospasm in some susceptible patients. Therefore, nowadays, it is no longer in wide use. Oseltamivir is considered to be a better drug. It is available for oral use. It is a prodrug. It is converted to active metabolite inside human cells. And oseltamivir is very effective against not only influenza A and B viruses, but also was effective, it was shown, against swine viruses and bird flu. If you remember, such infections were several years ago. And now about inhibition of attachment. The matter is that a lot of viruses have no special treatment against them. But it's possible to use the so-called pooled immunoglobulin against such diseases as measles, German measles, rabies, or poliomyelitis, using this pooled immunoglobulin. It contains antibodies against various viruses present in the population. So how does it is prepared? Pooled immunoglobulin is derived from plasma of 1,000 healthy donors. And the antibodies which are present in plasma of these healthy donors are directed against the virus envelope and can neutralize some viruses and prevent their attachment to host cell. Yeah, but of course it's better to use it before the onset of signs and symptoms. And I said you against which viral infections may be used for prophylaxis. For example, if such individuals are at risk of to be infected by these viruses. You see, there is also hyperimmune globulin specific against particular viruses, against hepatitis B, varicella zoster, and rabies. This picture represents some groups of antiviral drugs as a whole, which are used in clinical practice. You see, not all groups are shown here, but fusion inhibitors such as enfuvirtide, inhibitors of viral encoding such as amantadine, rimantadine, polymerase inhibitors such as acyclovir, zadovudine, and so on, protease inhibitors, and viral release inhibitors. So these groups of drugs you must know obligedly now, because they have been introduced into clinical practice not only recently but several years ago. They are used for a long time. They are very effective. You must know them. Yeah, and about some drugs which mechanism of action of which is not known well. It is decanazole at first. We read about. It is a drug which is effective against HIV, herpes simplex virus, and some other enveloped viruses. Then ribavirin. This is very interesting drug. It is very interesting drug. It inhibits RNA replication. It is thought to act either by altering virus nucleotide pools or by interfering with the synthesis of viral messenger RNA. But it is useful not only RNA viruses, it inhibits a wide range of DNA and RNA viruses, including especially respiratory syncytial virus. It belongs to RNA virus. Also may be effective against influenza viruses and may be effective against other viruses given here which cause infection of airways, respiratory tract. And about interference. Interference are divided into alpha, beta, gamma. Gamma interference, termed immune interference, is produced mainly by T lymphocytes as part of immunological response to both viral and non-viral antigens. We are speaking about alpha and beta interference are produced by B and T lymphocytes, macrophages, and fibroblasts in response to the presence of viruses and cytokines. So it is the step of immune response. Mechanism of antiviral action of interference. They work by inducing in the ribosomes of the host cell the production of enzymes that inhibit the translation of viral messenger RNA into viral proteins. So they act at the step mainly of translation, that is, formation of viral proteins such as DNA polymerase, RNA polymerase, and thus stop the reproduction of the RNA and DNA viruses. Interference binds to specific receptors on cell membranes. And now about treatment of the diseases by interference. Antiviral interference belongs to alpha group, and you may read about that this interference, especially alpha 2B, are very effective against hepatitis C. But also they nowadays are used in combination with other drugs against hepatitis C. May be useful for the treatment of other infections and unwanted effects. And now a few words about coronavirus, which is widely distributed now, widely spread now all over the world. Some information about, so you may read a group of related RNA viruses that cause diseases in mammals and birds. So coronaviruses include not only this COVID-19, but also some viruses which cause common cold, and also these groups of viruses also belong to. It looks like this. Shaped viral spike peplomeres colored red create the look of corona surrounding the virion, which is observed here. And this is how they look like under electronic microscope. And about structure of them. And a little bit about new drug. This drug is not new. It was synthesized in about maybe 5 years ago for the treatment of Ebola viruses. But nowadays it is tried to be used against coronavirus. It is called remdesivir. And it was shown that accelerates recovery from advanced COVID-19. You may read about mechanism is not so well understood, but likely it is inhibit synthesis of RNA. This is genome of these viruses. And about clinical trials with this remdesivir and how it can help to survive patients and to recover sooner. The end. Have you any questions?
No, Professor.
Goodbye.
Так.